In 2023, scientists reported that they'd been able to create synthetic human embryos using embryonic stem cells. Now, this establishes a model for important research on early embryonic development that could provide information on how to treat forms of infertility, to test drug safety on early development, and potentially to improve in vitro fertilization success. However, the current rules limit research on human embryos in a laboratory setting to only 14 days. So the question is, are these synthetic human embryos a less controversial method of studying early development since they don't have the same potential to develop into a new life in the same way that in vitro fertilization embryos are able to? So this presents our ethical dilemma in science for this week. Should research on these synthetic human embryos be restricted in the same way as normal human embryos produced by in vitro fertilization? In order to better understand what these synthetic embryos are and how they're created, we need to be able to understand and compare them through the normal process of reproduction and the assisted process of reproduction in the form of in vitro fertilization. Now, in normal natural reproduction, what's going to happen is that the sperm cell is going to come into contact with the egg within the female reproductive tract. Fertilization is going to occur, and then that fertilized egg is going to undergo a series of rapid cell divisions to result in a ball of cells. This ball of cells is going to reorganize itself into kind of a hollow ball, and then it's going to pass from the oviducts into the uterus. So about 7 to 10 days after fertilization has occurred, this hollow ball of cells is going to implant into the wall of the uterus and develop into the embryo. Along the way, it's going to produce some extra embryonic membranes by the interaction of these embryonic tissues and the maternal tissues. And the extra embryonic membranes is a fancy way of saying that it is going to establish the placenta, which is going to support and nourish the development of the embryo early on and ultimately the fetus as it develops further developmentally. Now, what happens in the process of in vitro fertilization, described here as ex vivo? Ex vivo is kind of outside of life or outside of the living body. In vitro fertilization is basically fertilization that occurs in a laboratory setting. And so eggs and sperm are going to be harvested from the donors or from the parents within a laboratory setting, within essentially a dish in the laboratory. The sperm and the egg are going to come together. We're going to have a process of fertilization that's going to occur. And then similar to what we see with natural reproduction, that fertilized egg is going to go through a series of rapid cell divisions, ending up in a ball of cells. That ball of cells is then going to reorganize into a hollow ball. And at that point, this hollow ball of cells would be implanted into the mother or a surrogate mother for natural reproductive purposes, or they can be frozen for storage and use further on. Ultimately, embryos created through the process of in vitro fertilization that are not used for reproductive purposes, they will be ultimately discarded or they can be donated for scientific research. In addition to these natural embryos formed by reproduction or the in vitro fertilization embryos, we're looking at these synthetic embryos down here at the bottom. And so what happens with the creation of a synthetic embryo is that we take some embryonic stem cells, so cells that are isolated from donated in vitro fertilization embryos. They go through a process in which they are kind of combined into a ball of cells, and these then synthetic embryos are going to reorganize into that same kind of hollow ball structure, and they can be studied in a laboratory setting. If we focus in on what occurs with these embryos, either a natural embryo or an in vitro fertilized embryo that are implanted into the mother or surrogate mother, is that following implantation, it goes through a process of embryogenesis. And so embryogenesis is a period of development that occurs after implantation and is generally completed by about 8 weeks of pregnancy. And it establishes the kind of rudimentary organs. It basically includes a primitive brain, a primitive heart, and other structures, so that by the end of 8 weeks of pregnancy, very primitive organs are in place within the embryo. And at that stage of development, it's going to convert from an embryo into a fetus. Now, the problem is that because the embryo is embedded within the wall of the uterus, it's surrounded by a placenta, it's very difficult, if not impossible, to directly study these early stages of development because basically the embryo is hidden within the wall of the uterus. And so there is an interest in learning about these early processes of human development so that we can improve reproductive success as well as study the effects of drugs on these early stages of development. If we focus in on what occurs to those embryos that are not implanted for reproductive purposes, we can see that they can be donated for research. And currently, there is a 14-day rule preventing development of the human embryos in vitro, basically in the laboratory setting, past a stage that's referred to as the primitive streak. This primitive streak establishes the orientation of the developing embryonic body. It basically designates what region is going to become the head region. It establishes the bilateral symmetry, so kind of a right and left side of the body. And so this is going to be kind of an important developmental stage. And now this in vitro development is limited to 14 days so that this process of growing an embryo in a laboratory setting ends before the central nervous system begins to develop. Now, this was originally proposed, this 14-day rule was originally proposed about 40 years ago when the technology was really, really primitive. These limits to research into early embryonic development limit our ability to answer important questions about early miscarriages or infertility or the effects of drugs on early embryonic development. In addition to that, the Dickey-Wicker Amendment within the United States bars government funding for research that creates or destroys human embryos. And this is placed as an amendment into the appropriation bills that fund the agencies associated with supporting research in the United States. Over the past 40 years, the science and technology for studying embryos in the laboratory settings has continued to improve. And as an example of this, we've got this artificial womb that is being used to study natural mouse embryos for an extended period of time within a laboratory setting. And so what happens is you take these mouse embryos, so embryos created by mouse sperm and mouse eggs brought together in much the same way that human in vitro fertilization occurs. We then put them into a solution of nutrients. We control the levels of oxygen and carbon dioxide, and so that we're able to support and nourish these embryos developing within this artificial womb. Now, what happens then is that these embryos can develop their primitive organs, but they don't have a true placenta which is surrounding them. And so there are potential limitations in that the placenta is going to have influences of the uterus that may or may not affect early embryonic development. Now, in the situation where we put these mouse embryos into these artificial wombs, we're able to grow the mice for about 11 days, which is roughly about half of the gestational development. And so it's the equivalent of making about halfway through the process of development. But we can't go beyond that with the current technology. In situations where we have used synthetic monkey embryos, so embryos created by fusing these embryonic stem cells together, implanting them into a surrogate mother, a surrogate monkey mother. We've seen the initiation of pregnancy, but they spontaneously terminate within about a week. Again, limiting the ability of these synthetic embryos to develop in a natural setting. And this may reflect, at this point, a limitation in the development of these synthetic embryos because we don't understand how to grow them further. When we take a look at studying embryos and focusing on human embryos in a laboratory setting, it is associated with a lot of controversy. Now, there are religious arguments against in vitro fertilization and research on human embryos, but we're going to focus in this talk about the effect of the Dobbs decision that overturned Roe v. Wade in 2022. In that decision, Justice Alito described abortion as an act that destroys potential life and the life of an unborn human being. Now, this argument could then be extended to in vitro fertilization embryos that may be discarded or donated for scientific research. And we've seen over the last year that states that had in place trigger laws that would restrict abortion once Roe was overturned, they defined the egg in many cases as an unborn child or as an unborn human being. And was created at the moment of fertilization, what they described as the moment of conception. So this could result in individual states extending these same principles that restrict access to abortion to limit research on human embryos or embryonic cells. And so while some states have specific laws to allow for embryonic research, other states could be in the potential to ban it. So currently we're in a situation where the laws and the guidance need to catch up with the science and technology that's available. In a variety of countries like Australia treats these synthetic embryos in the same way as natural embryos. And so they are requiring that researchers studying embryos in a laboratory setting obtain a license to conduct research, and they limit the in vitro development of even these synthetic embryos to 14 days. Other countries, such as the United States, the United Kingdom, and Japan, are currently treating these synthetic embryos differently since they can't produce a live baby. Now, the rules associated with studying embryos in the laboratory setting are established by the International Society for Stem Cell Research, and they issued new guidelines in 2021. They basically called for potentially relaxing the 14-day rule, but only relaxing that 14-day rule after gaining public support. Then research that would go beyond these 14 days would require approval by a committee of specialists and laypersons that determine that the research is justifiable. Regardless, though, they're still prohibiting the transfer of human embryo models that would then be implanted into human or non-human uteri. In order to consider the ethical dilemma in science for this week, should research on synthetic embryos be restricted in the same way as in vitro fertilized embryos that have the potential to develop into a living organism, we need to consider a variety of questions. First off, should synthetic embryos using human cells be created for research? Should the restrictions on embryo development be based on age, as it is now with 14 days, or should it be limited on the potential to become a viable offspring? If these synthetic embryos are so similar to normal in vitro fertilized embryos, should these synthetic embryos be protected by the same rules, or at least rules similar to those protecting the development of in vitro fertilized embryos? And then finally, should synthetic or in vitro fertilized embryos be allowed to develop beyond the 14 days in the laboratory setting so that we can learn important details about early development in order to develop better technologies for treating infertility, better drugs for treating maternal conditions that are not as harmful to the developing embryo, as well as learning more about the process of human reproduction. So thank you for listening to this week's Ethical Dilemma in Science. As always, please see the description below for additional information and references. Thank you, and have a great week.